Thank you very much for attending the session, and thank you very much to the conference organizers for the opportunity to speak. So today I'm going to talk about sort of a long-running program we've had on really focused around identifying disease-causing variants in autoimmune disease. As the technologies have evolved, I'll show you how we've sort of embraced some of these different technologies over time to get more and more high-resolution information. So I do have to give a little bit of information on sort of how autoimmune disease happens. I mean, most people have a pretty basic understanding of it, but really your body has this amazing way of making sure that it's making immune cells that will attack the foreign antigen, but actually won't attack your own cells through these tolerance checkpoints. But like anything in nature, there's a lot of ways that these mechanisms can break down. And so what happens is some cells escape the become autoreactive and escape these checkpoints. These can get activated over time, and then they proliferate and develop into these low rogue cell lineages. And then this can lead to full-blown autoimmune disease. So what we can do is we can sort of, as early as we can look back and say, what are the mutations that are actually causing these things to escape immune tolerance? And it can be found either germline or somatic driver mutations. Traditionally, people think of germline mutations, but hopefully I'll make the case that somatic mutations are really important in this as well. And the other thing we can do is if you have someone with autoimmune disease, you can come along and say, what is the actual disease doing? Autoimmune disease is a bit of a grab bag of diseases. There's a lot of different ways that these diseases manifest in people. So you can actually look at what they're doing, for example, pre and post treatment and things like that. So how do you actually find driver genes in autoimmune disease? So there's germline mutations. That's something you're born with. That's sort of falls into your classic kind of rare disease analysis workflow. It can usually is something that you inherit from a parent, or you can inherit multiple faulty alleles from parents and things like that. But that's a little more straightforward, and we do that. But I'm going to focus a little more on the somatic mutations today. So when you think of somatic mutations, you usually think of cancer. But again, hopefully I'll be able to convince you that they're actually really important in autoimmune disease as well. One additional challenge with autoimmune disease, with cancer, you have a tumor that you can usually get a fairly high level of purity and then a control. So it's a sort of one-to-one sampling. With autoimmune disease, however, you have a lot of different mix of cell types. And usually it's one of these cell types that is actually driving the autoimmune disease. So you have this additional layer that you have to sort of deconvolute the type of immune cells to figure out which ones are accumulating the variants. And that's where single cell is quite useful, obviously. And I'll walk through a few cases, just very high level, obviously, but I'll walk through first case is just sort of a WGS and single cell 10X. Then I'll walk through 3 cases of somatic mutations over the last sort of 5 years we've covered, which use GNT-seq, GT-seq, they're different, and single cell DNA-seq, a really new technology we're kind of excited about as well. So we have, for about a decade, we've had sort of a classic precision medicine program in genetics, basically finding disease-causing variants in autoimmune disease. This was essentially had some lucky enough to work with some very forward-thinking clinicians at the time. About a decade ago, we realized the importance of genetic variation in autoimmune disease, and they reached out to me and a fellow bioinformatician, and we put together a big program to do that. So that thing's still churning along, and now is in the running in hospital settings, and also has a research arm as well. So that's great, and is I guess what you sort of more think of as a traditional personalized medicine program. Here's just an example. This is a recent case where these people were on sort of this diagnostic odyssey for about 20 years. No one ever could figure out what was causing their disease. So essentially, we did WGS on them. He got referred by a smart clinician to us, and luckily, we were able to find a new driver gene. And that was sort of your standard kind of workflows. We did our usual clinical workflow. We then generally have to do a CRISPR mouse to recapitulate the phenotype to prove that the causal variant is what you think. And then additionally, what we're starting to do now is 10x single cell sequencing on the other side to actually see really what's happening. So this one, we were lucky we had pre and post treatment where the treatment worked beautifully. No one would have given them the treatment, but based on the mutation, they changed the treatment course and it cured it beautifully. So we had nice samples from before and after, and we went through and we did sort of all the standard 10x single cell kind of things with normalization and cell assignment with azimuth. Increasingly, people are using site-seq as well. You can use really well combined with 10x to actually annotate your immune cells and your DE. And then you basically take your patient phenotype data with your omics data, and then you can really dive deep into the mechanism of what's happening. Allows you to do things like this. This isn't published, so but hopefully very soon. And you can do your UMAPs and your volcano plots and your enrichment plots and all that sort of stuff. But the work I'm going to talk a little more about is the somatic mutation. So I was fortunate enough to do my PhD with a very eminent immunologist, Chris Goodnow. And he had this idea, this is from 2007, that actually autoimmune disease developed in this sort of multi-step pathogenesis mechanism, similar to exactly how cancer, it's a single cell that accumulates mutations, it gets a competitive advantage and expands over time. And while this was great in theory, the technology didn't really exist to prove it. Part of my PhD was trying to figure out how to actually tag molecules prior to PCR amplification and doing all these crazy things. But at the end of the day, technology just wasn't quite there. So essentially, in a nutshell, you have your antigen and your antigen receptor. And then basically what happens, there's different ways that this process can break down. You can either have growth limiting mechanisms or growth overriding mechanisms. And this will result in one of three outcomes. The first outcome is the positive one. The foreign antigen's cleared from the body, the cells will die off, and it'll either be go into memory cell or be the end of it. So that's what you want to happen. But if you get mutations in the control pathway, then you get this relentless growth. So you essentially get cancer of the immune system. Or if you have a self-antigen that can't be cleared, then you again have this relentless stimulation resulting in autoimmune disease. And don't worry too much about this, but this is just to illustrate that even at the time, we understood it wasn't a simple one mutation would occur and it would just unfold. It's quite a complex process and a different combination of factors coming together. So the first case, our proof of principle, was led by PI Joanne Reed, who's at Westmead in Sydney. This one we basically wanted to find lymphoma driver mutations in this rogue cell lineage that they knew was driving this autoantibody expansion. So for this, we use a technology called GNT-seq. Essentially what that does, you would basically take cells that were already sorted, and then you would split them into mRNA and genomic DNA libraries, and then you would essentially get one cell per 96 well plate. And that would essentially be what I would analyze. So the nice thing about this technology is the experts in the lab who knew exactly what the cells were and what the lineages were. So then you could go through, and for me, it was quite easy because all I had to do was just sort of expand the somatic variant detection and just say, what are the mutations that are in these 20 cells that we expect to have them and not in the rest of the cells? So it's fairly straightforward. And luckily, we were able to crack all 4 patients we looked at. Everyone had at least one driver and one had 4 drivers. That were known mutations in lymphoma, which was great. They also simultaneously did the B cell receptor sequencing, so we could actually see the clonal expansions and how the mutations aligned with this. So it was very powerful to put the 2 data sets together, and we had one patient we had about 5 years of data for, so it was quite a powerful way to really watch this rogue lineage expand over time and accumulate mutations. So this really showed us that we were seeing these rogue mutations. They were unique to these expanding lineages, and it really corresponded beautifully to the B cell receptor expansion. And this was published in 2020, if you want to look more into that. The next one was a case. This is a PhD of this girl, Clara Young at Garvin. She essentially, this case, it was a virus-induced autoimmune disease, and in this case, we used a technology called GT-seq, which is essentially amplicon sequencing of thousands of cells, essentially, or hundreds of cells across a targeted panel. In this case, we had about 600 known lymphoma genes that they maintained and generated panels for. And really, the analysis at the end of the day was very similar to G and T-seq. But in this case, there was 4 patients. I could only crack 2 of them using this targeted approach. So luckily, I convinced them that there might be something else going on, and they basically took their 2 lineages, the control and the rogue lineage, and amplified it in the WGS. And actually, that proved very beneficial because we found these were the 2 small mutations we found. This actually ended up being a large deletion, resulting in a new gene fusion, somatic gene fusion. The other one was actually a trisomy. So they all are known to be involved in lymphoma, but we really had to go beyond sort of looking just at single bases for this sort of thing. So again, we could do the look at the clonal lineages, and you can see different levels of expansion, quite interestingly. This was a preprint you can look at, and this to us sort of highlighted that we have to look up for lots of different types of drivers. It was exciting because we really was a very clear link between the virus and autoimmune disease, which was exciting to see. And then the other thing that you realize when you start to look at these things is once these clones have expanded and accumulated these mutations, it doesn't actually matter if the stimulus is still there. So this was Hep C was the virus in this case, but even if you remove the virus, the clones are still going to be there expanding and driving the disease. And this was just accepted last week in Immunity, so there'll be, you can read that paper if you want. And then the last case I'll talk about was refractory celiac disease. That's a very advanced form of celiac, again, where essentially even if you remove gluten from the equation, they still have the syndrome essentially, and it usually progresses to cancer from that point. So it's not that common, but when it happens, we wanted to use it as a test case for whether in T cells this time. So for this, we use a single-cell DNA sequencing technology called Mission Bio. It works on a small targeted panel. It's not a whole genome-wide application. And it also allows you to do the immune cell protein markers as well. So you can do that simultaneously, which is great. So at the end, you get sort of your target amplification. You'll get your DNA and your DNA FASTQ for each cell, and then also the immune cell marker FASTQ as well. So it's quite powerful that way. This is a bit harder though, because it has its own pipeline. It output a big HDF5 that was sort of SURAT ready object, but then it'll give you this giant VCF of 5,000 samples. Essentially each cell is a sample, and it was quite hard to work with. There was hundreds of variants called, and it had its own sort of black box filtering where it did what it thought was important, but we wanted to build our own around this because we didn't want to just trust the black box solution. So we did a lot of time annotating things, an excellent postdoc, did some work on developing sort of this combination supervised and unsupervised learning approach using the marker data to annotate your cells. And then he did this sort of customized, custom supervised approach where he sort of weighted the really important markers. There's lots of markers to tell what kind of immune cells are what. And in the end, this changed some cell assignments, but it was actually very useful for finding additional doublets and dead cells that were actually making the data a bit noisier. So that was where it was. And then I came in and did a lot of the optimization of the filters and things like that. And then we wanted to find things that were enriched in individual cell types. So we sort of came up with this idea of iterate over the cell types and then compare each cell type to all the rest and look at the variant frequency and then develop odds ratios and Z scores and that sort of thing. So that sort of gives you, it generates reports, this output. This one's for CD8s, and you can see the frequency of the variant in CD8 cells versus the other, and this one obviously is very enriched. But the thing with this technology is you can't trust 100% of the cell annotations because some of them are going to be wrong, and that's just the nature of single cell data. So you just have to look for enrichment. You can't sort of take this black and white approach that all the variants should be in this type of cell. So that's where the statistics were needed. You can visualize things. This is just, again, don't worry, the resolution's terrible, but this is just showing how high priority variants by cell type. And on the one on the left, you see there's no real pattern. And then if you look on the right here, you can see there's some cell types that are enriched here. And then you can do, this is one patient that had 5 driver mutations, and you can do upset plots and see, we're looking at how we can use this to sort of infer clonal lineage as well. So essentially, as I mentioned, we did this on about 25 patients now. We annotate all their individual cells. There's a preprint you can read. And lo and behold, it was extremely successful. We found, yes, these refractory celiac patients do accumulate somatic drivers. In fact, 75% of the 9 of the 12 patients we looked at had drivers. And again, we couldn't rely fully on the cell annotations. So just wrapping up the last thing, this is what we're looking at here. This is the advanced refractory, so that's RCD2, the most advanced form of it. And what you're looking at, the bars at the top are the cell type annotations, and then the red is the mutations, and of the 4 mutations in the top and 5 mutations on the bottom, and then the shade of red is whether it's homozygous or heterozygous. So the top one you can see they're pretty much in that rogue cell line. There's lots, and they co-occur. Second one, you can see that they are again in the rogue cell line, but they don't co-occur. There's 2 different sort of mutation occurring there, pathways occurring there. And then this is the intermediate ones, RCD1, and you can see that some of them are starting to accumulate these mutations, a few of them weren't. And interestingly, the one at the top that had 5 driver mutations actually we think was misclassified as RCD1 and should be RCD2. So it really shows there is an accumulation of these mutations over time. So I'll wrap up just by showing you how we leveraged a number of different technologies to do a personalized medicine approach for finding autoimmune disease drivers, both germline and somatic. We had to develop a new pipeline for the single-cell DNA and protein cell markers. And what this hopefully will lead to is, imagine, just like anything, the earlier you can detect things, the better. So that's sort of the motivation there. I want to thank everyone I thanked along the way, then Associate Professor Simon Jang for the germline work, Chris Goodnow for the somatic work, my postdoc, 10X work, funders and providers, and then one last little plug. If you like the tropics and try to remember 3 days ago when it was sunny and beautiful and not the last 2 days, I live in Cairns, Australia, and I've got a postdoc position and some PhD students if anyone wants to talk to me about that. Thanks very much.
Thank you very much for your talk. Any questions or comments? Okay, I have one question.
Sure.
I think the mutation analysis is very popular in cancer research. I agree that it is important to apply such kind of approach to autoimmune systems, autoimmune diseases. So what are the most difficult problems or most difficult?
Yeah, it's a great question. Specific to autoimmune disease. I mean, the one thing in particular was that, cancer, you usually have one tumor, sometimes multiple tumors, you'll have a primary and a metastatic, but programs are designed to handle sort of a smaller number of samples, whereas we would sometimes have 100 cells, and so we would have essentially 20 tumors and 80 normals, and so I actually had to play around. Luckily, new version of MuTech4 just came out, MuTech2 in GATE4 that actually allows you to have an infinite number of combinations to do that. So we're exploring using that, but essentially most of the programs aren't well set up for that scenario of multiple samples of essentially what you call the somatic ones that you're looking for and the controls. So it definitely is a challenge. And a lot of them consider things like tumor purity and things, which isn't relevant in our case as well. So it's a few different challenges. So we have a combination of what we could get from the cancer space because it's so advanced and there's so much going on. And then when there's no solution, we build our own, essentially.